Welcome to our third week of the Space Synthetic Biology Festival at the European Society of Synthetic Biology. Today, we've got a very esteemed guest. We've got Dr. Daniela Billi, who is an associate. She is a professor of environmental microbiology at the University of Rome Tor Vergata, a leading expert in astrobiology and molecular biology. Her research focuses on how cyanobacteria survive in Earth's most hostile environments, such as hot and cold deserts and Antarctic subglacial lakes, and how they adapt to stimulated space and Martian conditions. Dr. Billy has served as principal investigator for major space experiments on the International Space Station, including the BOSS in biofilm organisms surfing space and BioMex, Biology and Martian Experiment projects through the EXPOSE R2 mission. She also holds a scientific responsibility for collection of roughly 200 desert cyanobacteria strains of the genus Chroococcidiopsis. Beyond the understanding the cellular limits of life, her work actively bridges the gap between Earth's desert and deep space by engineering cyanobacteria to develop sustainable off-world life support systems for future human outposts on the Moon and Mars. So please welcome us today, dear audience and recorded audience, a very esteemed guest, which I'm very happy and proud of having here today. We are honored of having you here today.
Thank you for this nice presentation and also for the invitation. So let me share the screen now. Okay. Okay. So as you mentioned, we will do this a trip from desert to space. And I will say also a story of this desert cyanobacteria belonging to the genus Chroococcidiopsis, on which I started to work a long time ago. Yeah, so this cyanobacteria has been discovered in the Dry Valley in Antarctica that were considered to be let's say, a place too extreme for life. And this cyanobacteria has been discovered taking refuge inside the rock, as well as they can survive in hot desert like the hyper-arid desert in the Atacama Desert in Chile, where they live inside the rocks or between the rocks and the soil. And if you look at these extreme environments of an Earth, there really seems to be a Martian landscape. So these places are not very different from the picture that the rover Curiosity has been sending to us from Mars. And life in desert on Earth is also saying us what could have been happened on Mars when it changed from being warm and wet to dry and cold as it is today. So we can speculate that if life colonized the terrestrial environment of Mars, when the habitability condition on the surface has been lost, an hypothetical microbial life form could have taken refuge inside rocks, like it happens on Earth. And so it is meaningful to search for life currently not on the surface of Mars because the conditions are too extreme, but under the surface. And so, as I said, these desert cyanobacteria were first discovered in 1976, so a long time ago, in the McMurdo Dry Valley in Antarctica, by Imre Friedman and his wife Rosilio Campo. And they published this in Science, in the journal Science, and their publication on endolytic life in this extremely dry and cold desert on Earth was concluding, saying that if we are looking for life on Mars, we should look under the surface. And this because this was the year when the rover Viking failed to give evidence on the existence of life on Mars. So at that time, this was quite a very hot topic. And since then, desert cyanobacteria has been used as a model system in astrobiology. And here you can see a graphic novel from NASA where it is a that is let's say a drawing about Himmler Rosili discovering these cyanobacteria in Antarctica. And since then, this cyanobacteria has been used as a model system for astrobiology with the aim to find life beyond Earth with a particular focus on Mars. And actually, this is me after my PhD. I spent 1 year with Imre Friedman in Tallahassee, Florida. And actually, as you mentioned at the very beginning, Imre has realized, along with his wife, a huge collection of cyanobacteria of the genus Chroococcidiopsis. And after his retirement, he transferred to me at Vergata University this collection that has been really the result of more than 40 years of traveling in very extreme desert on Earth. So it has been really an honor for me to take care of this collection. And over the years, of course, now it's more than 30 years that I'm working on this desert cyanobacteria, with my PhD supervisor, Maria Grilli Caiola, we started to understand how these cyanobacteria can survive extreme desiccation. And we figure out that since this cyanobacterium is unicellular, but is able to perform binary and multiple division. So usually you have a multicellular aggregate where actually you can have a dead cell in response. These has been dried for 6 months. So it is evident that some cells die, whereas others can survive. And when you transfer them back to radiation, so you put them back to life again, they enter multiple fission, and so you have very big multicellular aggregate taking place. So these were really the very first let's say, studies that we performed. And we were also very curious to understand what was happening to survivors. So we looked at the DNA damage using an assay that is named tunnel assay, where actually you can detect DNA breaks, single or double breaks, using the incorporation of nucleotides that are linked to a fluorescence label. And so we could see that actually that cell has a lot of these spots, meaning that there are a lot of DNA damage. And also we saw that also the pigments, the photosynthetic pigments were lost, whereas we could see cells with just a background signal, and also cells that were maintaining the photosynthetic pigment, and cells again with the strong dots-like signal that were dead without photosynthetic plasmid, so photosynthetic pigments. So it was clear that some cells were dying, whereas others were actually able to protect their subcellular integrities, especially also the DNA. And also a long time ago, we performed a PCA-based assay to detect DNA damage. So we extracted DNA from dried and control culture of Chroococcidiopsis. We used some small random primers, and the rationale is that these primers will amplify the DNA because they will find the complementary region. Whereas if there is DNA damage, in some region of the DNA there will be the possibility of the annealing, but also of the PCR amplification to perform the DNA amplification. So if you look this on the gel, you will see that some bands are missing, and it is what we saw. This is this polymorphic amplification of liquid culture. And if we look at the dry, we already see that some bands are missing, so some damage has appeared. And the damage is also repaired because in the rewetted, upon rehydration and extraction of DNA and performing this PCR amplification assay, we saw that there is the typical bands profile. So damage is indeed occurring, but cells are able to repair this induced damage. So over the time, we also made some comparison to better understand how these cyanobacteria can survive desiccation, making a comparison with the desiccation susceptible cyanobacterium like Synecocystis PCC6 3, sorry, there is a mistake, 6300 or 08. And we checked we extracted the protein and we checked it to carbonylation. That is an easy, it is a kind of oxidation damage to protein that can be easily detected because you just buy antibodies against this carbonylation. And we saw that in dried sample of Chroococcidiopsis, protein are not so heavily carbonylated, so they didn't undergo oxidative damage because the signal is comparable to that in liquid culture. But if we look to what is going on in Synecocystis, you see that in dried cell there is a huge amount of oxidation. And of course, all the protein are damaged, including those that are involved in the DNA repair. So this is something similar to what has been also published for the Inococcus redurans, where actually the capability to avoid oxidative damage is really very important to survive not only desiccation but also radiation. And so we with the idea that Chroococcidiopsis or this strain of Chroococcidiopsis has an important and efficient system to avoid oxidative stress, we looked at the genome, and indeed there are a lot of genes involved in the antioxidant defense. So compared to Synecocystis, we have, for instance, 3 superoxide dismutases, whereas Synecocystis has only 1, and there are also a lot of catalases and peroxidases. So for sure, at least this enzymatic defense is really very amplified and important, and we think that it contributes to survive desiccation, and as we will see, also radiation, because also radiation is inducing damage in an indirect way through the production of super or Ross species. And another point that we also investigated is that it's very well known that anhydrobiosis, that is the capability to survive the complete water removal, is often associated with the water replacement theory, in which organisms that can survive desiccation without without the differentiation of akinase of spores, so organisms that can survive, let's say, in a vegetative state desiccation, usually they produce a large amount of trehalose, but also sucrose if they are photosynthetic organisms, and these two disaccharides are replacing the water molecules so that they prevent cell membrane, but also protein, to undergo all the problems that are linked to water removal. That is, let's say, a stress that is lethal to the majority of the organisms that we know. And we also looked at the genome of desert strain of Chroococcidiopsis, and actually we identify several genes that are involved in the synthesis of sucrose and trehalose using different precursor. So it is like that when desiccation is occurring, desert strain of Chroococcidiopsis can use a different precursor to accumulate sucrose and trehalose, and so avoid desiccation induced damage. And as I said, usually organisms, microbes that are extremely desiccation tolerant are also extremely resistant to radiation. So if you extract the DNA, for instance, from a control liquid culture and you put the genomic DNA on gel, you will see a band. And then after irradiation with very high doses of gamma rays, like 5 kilograms, that is really a little dose for most of the microbes. You see that the DNA it's all fragmented. So on the agarose gel, you see just a lot of smears, so a small fragment. And if you leave then the time to repair after 12 and 14 hours, you see that the DNA is recovered. So this is something very similar to what has been also shown for the Inococcus reducens. And when we tested the extreme capability to survive ionizing radiation, we reached the doses as high as 15 kilograms. That is really a huge dose of ionizing radiation. And here you have 4 different desert strain of Chroococcidiopsis from different deserts, from Antarctica, from Negev, from Sinai, from Mexico. And you see that more or less they are all extremely resistant, especially if you compare the survival with that of E. coli or Synechococcus, that they die very quickly after very low radiation. And this kind of radiation resistance of Chroococcidiopsis, it's really putting this Cyanobacterium, or at least desert strain of this Cyanobacteria, in the top 10 of microorganisms that are extremely resistant to radiation. And these experiments were also performed under the same experimental setup, which is also important. And here you see the doses for the D10 doses. So maybe it's a little less resistant than the Inococcus reducens, but still these Chroococcidiopsis strains are really being also photosynthetic organisms, where actually they produce also oxygen, and this will exacerbate the oxidative stress that is also associated with radiation. They are really extremely radio resistant, we can say. And so as I promised, we will go from desert to space. And I want to point out that being desiccation and radiation tolerant, it's really a prerequisite to be exposed in space. And this because the first experiment, let's say astrobiological experiment, dealing with investigating the limit of life as we know it, has been performed using an exposure facility of the European Space Agency called EXPOSE, that is installed outside the International Space Station. And in this facility, and if you expose organism to space condition, then space will induce space vacuum will induce desiccation. So being desiccation tolerant is really important. And if you expose the cell to Martian simulated condition, utilizing the fact that the International Space Station is outside the atmosphere, so there is no protection against UVC, and also there is a very tiny shielding against cosmic radiation, because it is almost out of the Earth magnetic field, so you can simulate the Martian condition in low Earth orbit. So, but Martian has, the Martian condition is also including low pressure, so like 600, 800 Pascal, and also low pressure will induce desiccation. So in this experiment, as you were mentioning at in the introduction, organism has been exposed to space for 1 year and a half, to space and Mars-like condition. And so, and the period was already almost 10 years ago that we were involved in this experiment, that are very much fascinating, because your sample is launched during a space mission. It will reach the International Space Station, and then of course you have to wait for the astronaut extravehicular activity, so that they will install outside the ISS the ESA facility with your sample. They will stay outside there for 1 year and a half, and then the astronaut will take them back inside the ISS, and then they will return on Earth under the seat. In this case, this was under the seat of Team Peak, that was the command of this mission. And here you see two of these two pieces of these facilities after being exposed in space for 1 year and a half. And if you go to YouTube, in the Italian Space Agency channel, you will also listen to the description of this mission made by our astronaut Samantha Cristoforetti, that was inside the ISS for the first time, while these two experiments were outside the ISS. So this is a school of astrobiology, so it is an outreach for the Italian young researchers. And so the two experiments in which desert cyanobacteria has been used and tested in space were both biofilm organism surfing space and biomax biology and Max experiment that has been selected by ESA and funded by the Italian Space Agency. And in the overall feature of this space experiment is that cyanobacteria, desert cyanobacteria of the genus Chroococcidiopsis, has been air dried on Earth. Then they were implemented inside the facility, exposed outside the ISS in the dried state. So while dried, they accumulated the damage because they are exposed to space vacuum, to ionizing radiation, and also to Mars simulated condition. So really extreme condition. And since they are dried, damage are not repaired, so they are only accumulated. And once back to Earth, we investigated the biomarker stability, because astrobiology is also always looking for the detection of biomarkers and see how space and Mars-like condition can alter the detectability of biomarkers. And then we radiated back these dried space exposed samples to look for survival. And so to look for their capability to repair the damage that were accumulated while in space. So to give a better look to the biofilm organism surfing space experiment, biofilm can be obtained by plating cyanobacterial cell on agar, and the biofilm, when organisms are shift from liquid to liquid culture to the possibility to grow on the top of surface, they grow in layers, and they are also very rich in polysaccharides. And so the reason why, when biofilm were exposed outside the ISS to space and Mars-like condition, we scored the survivors is that the cell on the top of the biofilm will die. So this is an image of the confocal microscope where you can see layers after layers decomposition of the biofilm. So going from the top to the bottom of the biofilm, we saw that on the top the cells are dead, whereas on the bottom they are alive. So this because UVC mainly, it's really lethal, and but it is a kind of radiation that is not very much penetrating. So top cells were killed, but they also offered protection to the cell at the bottom of the biofilm. Whereas the ionizing radiation that are really very much penetrating, the dose is not so high. So in here outside the ISS, it was like 0.5 gray. That is nothing compared to the kilogray of ionizing radiation that this cyanobacteria can cope with. And also because the cells were dried, so less oxidative stress is produced because there is no water. So also radiation tolerance is even increased when the cells are dried. On the other experiment, Biomax, Biology and Mars experiment, liquid culture were mixed with the regoliths that were, simulants of Moon and Mars regoliths. So that the survivors that we scored after that the sample were exposed outside the ISS and then retrieved back to Earth, we saw some survivors, and these were the cells that were shielded by the regolith grains. So here you see the cyanobacterial cells in red, and these gray things are the regoliths. So the regoliths is always enough to protect the cell from UV radiation. And as I said before, the general investigation is first, once that we had the sample back, is to look for the permanence of pigments. So we went to the confocal and we looked that actually there is some damage and so some loss of signal, but still there is some detectability. DNA can be, as if we see with the amplification with the PCR, depending if the cells are protected or not, the DNA can be differently damaged. So the damage is occurring, but the cell, since they are then alive when rehydrated, this kind of damage that we detected can be repaired. And also we look at the Raman signal using the Raman microscope, using the same setting that will be on board the ExoMars rover, named also Rosalind Franklin, and we see that in dried and also exposed to space condition, the signal of carotenoids is still present. And this is important because carotenoids are not only associated with the photosynthesis, but also non-photosynthetic pigment have carotenoids. So this is also important as a suitable biosignature. And since we noticed that DNA actually was damaged, but that the cells after exposure to space and Mars-like condition were actually able to divide, we obtained what we call a space-derived Chroococcidiopsis strain from a piece of sample that was exposed to Mars-like condition in space. And we sequenced the genome of this space-derived strain, and we made a comparison with the ground reference, so just the normal strain that is part of our collection. And we saw that the number of variants were actually not increased in the space-derived strains compared to the ground reference. So the typical mutational rate is not increased. So this means that the damage that were accumulated, the DNA damage that were accumulated while in space were actually properly repaired under Earth conditions, so basically in our laboratory. So we are very much interested in better investigating this capability to repair DNA damage accumulated in space, but we want also to understand the effect, the capability to repair DNA damage under non-Earth condition. So we have been selected for an experiment that will be performed on board the Space Rider, that is an unmanned vehicle of the European Space Agency that will fly in 2027. And what we will do, we will have a Chroococcidiopsis strain with a plasmid, synthetic plasmid. So here we are also embracing synthetic space, synthetic biology. We will dry the strains. We will irradiate it with a dose that has a significance for space biotechnology. Let's say we will simulate the dose that is accumulated during a transit to Mars. The sample, so with the plasmid, the irradiated sample, will be radiated in space, and we will wait a few days to allow the DNA repair, and we will fix then the sample, and the sample will retrieve it back to Earth, and we will investigate the genome sequence, and also we will sequence also the plasmid, and we will see if the synthetic plasmid will be as the original one, meaning that the DNA that is accumulated is efficiently repaired also in microgravity and under space radiation. So this is important. This is a very first step towards space synthetic biology. And meantime, we are performing ground activities to get ready for this experiment, and we are using the simulated microgravity in the laboratory. And so this is ongoing. And of course, it is to move forward, because as I said before, the first experiment, the first astrobiology experiment in space has been performed using dried metabolic microorganisms, that is important because we could expose them to space condition that, as I said, there is vacuum, so of course you cannot think about metabolic activity in the vacuum, or under Martian pressure that is also reduced compared to Earth. So this first experiment has been really very important to understand the limit of life as we know it, and also the stability of of macromolecules that we are using as biosignature. But it is also very important to see the limit of life in space and test it using a metabolic active organism. And this is very challenging because we need bioreactors that must be miniaturized, especially if we want to use CubeSat or small satellites, since performing experiment in space outside the ISS, it's very expensive, and also considering that the ISS will be decommissioned in a few years. So it's also important to look for other platforms. And CubeSat or small satellites are important because these are more cheap experiments to be performed. And being a CubeSat, of course, you cannot have your sample back. So you need also not only to find a way to keep your cell alive while in space, so you need to have pressure, temperature, light, everything controlled, but you need also to know from Earth what is going on in space. So you need real-time monitoring methods that can be easy to be done in the laboratory, but then you have to miniaturize this, then it's more complicated. And we have been working lately in a European project in which we transformed the desert cyanobacteria with a plasmid that was able to emit a luminescent signal once that the DNA was damaged by ionizing radiation. And the signal was detected using a lab-on-a-chip technology that has been developed for space application. And this work is just now in press. And so this is also another step towards knowing what is happening in space and what kind of response is going on. So this is very important that we can monitor this from Earth. And in the near future, we have been recently we have been selected by the European Space Agency in a pool of projects that could be performed on the moon and on the moon surface. And this is also very important to astrobiology because the environment is more extreme. There is no atmosphere, so there is no protection against radiation, there is no magnetic field. So the moon, even if it is not of interest from an astrobiological point of view, it is important because it is offering a more extreme environment to test the limit of life as we know it, and so also contribute to define the boundary condition of other planet, the exoplanet and other world where we are looking for the possibility of life. And so being on the moon surface, of course, and not being sure of having the sample back, we have been working in developing a real-time monitoring methods to know about life and death of cell while on the moon, and we are developing this together with Kaiser Italia, and we tested the different molecular probes that maybe change from being non-fluorescence to fluorescence when they are modified by cellular enzyme. And of course, this fluorescence signal has to be detected in a miniaturized experimental unit, and the signal has to be transferred to Earth. And so these experiments are currently going on, and they are funded by the Italian Space Agency in the frame of the Extremum project, in which we are also looking to biomolecules, and so we are also paying attention to biosignature detection. And desert cyanobacteria are, as I said at the very beginning, they are surviving in desert because they are taking refuge inside the rocks or between the rock and the soil. So they are endolytic, chasmoendolytic, cryptoendolytic, or hypolytic. But these organisms are photosynthetic organisms. So light, of course, is not, and even though these rocks are translucent and some light is passing through, according to the rock composition, there are conditions in which the visible light is very much attenuated and the cells are reached by infrared light. And this is very interesting, not only to understand the life in desert, but also to contribute to the search for life on exoplanet. And I will explain why. So what is happening on Earth is that the cyanobacteria are the only photosynthetic organism that can transform chlorophyll A in chlorophyll F. So if you look at the absorbance spectrum, you see that when they are grown under far-red light, they produce a new kind of chlorophyll D and F that are able to absorb beyond 700 nanometers. And this is very important because as far as we were at school, we studied chlorophyll, the oxygenic photosynthesis in the visible light. So the possibility for photosynthesis beyond the visible light is very intriguing, not only on Earth, but also to search for life on exoplanet. In particular, I'm talking about exoplanets around M stars, because here you see the emission spectrum of our Sun that is very much enriched with a strong emission in the visible light, whereas M stars that are very abundant in our galaxy have a very tiny emission in the visible part of the spectrum and a more intense emission in the infrared light. And since oxygen is a biosignature that is relatively easy to be detected in the atmosphere, and pigments also are a biosignature because there is the surface biosignature due to the reflectance of the pigments. So the possibility for oxygenic photosynthesis is very intriguing. But of course, when we look for a potentially habitable exoplanet, is not only the possibility of using light, but there are all other conditions that has to be considered. So not only because these stars are of course, if there is no magnetic field and if the atmosphere is very tiny, organisms must face solar flares, and also ionizing radiation. So in a study that is going on in my laboratory in the frame of a project that has been supported by the Italian Space Agency, we are investigating how cyanobacteria that are able of red acclimations or are able to use infrared light to drive photosynthesis can, let's say, survive UV radiation, and we are simulating conditions that are occurring on the surface of two exoplanets that are really two important targets for astrobiology, that is TRAPPIST-A, that is one of the three planets in the habitable zone of the TRAPPIST, and Proxima B, that is also an habitable planet. And we are simulating the flare that are arriving on the surface of these two exoplanets, and we are also recording the survivability using a prototype that could be used also in space. And of course, we're using conditions that are, let's say, simulating subsuperficial condition in aquaplanet, and I can anticipate that we actually found the possibility of these cyanobacteria to survive high doses of UVC, comparable to flares that are typical of these M stars. And to move from this astrobiology data to a sort of applied astrobiology, I want to stress out that all that we are investigating on the possibility of cyanobacteria to survive space, Mars-like condition. Now we have seen also exoplanet exoplanetary simulated condition in the laboratory. All this is very important to astrobiology, but has also an application towards space biotechnology, towards space synthetic biology, because the fact that we know now that we can move from the laboratory to space dried cyanobacterial cells. This is important because they could be transferred from Earth to the Moon, maybe in the future to Mars. They can be rehydrated, and also being photosynthetic organism. We did some experiment in my laboratory where actually we obtained the nutrients from simulants of Mars and Moon soil. And since desert cyanobacteria of the genus Chroococcidiopsis are not able to fix nitrogen, but they have the gene to use urea as a nitrogen source. We use synthetic urine to support their growth. And of course, they use CO2, so that is either present on the Mars atmosphere or produced by a human crew, for instance. So we can cultivate desert cyanobacteria using what it is called as in situ resources. So we can use regoliths, we can use urine, we can use CO2, so we can use them in a sort of bioregenerative system, because the biomass that we obtaining cultivating cyanobacteria using in situ resources can be used as feedstock to support the growth of bacteria that maybe are more suitable for right now as a chassis for biotechnological application, but being bacteria, they are heterotrophic, so we know that to grow bacteria in the lab we need very rich media, and of course we cannot transfer this media on the moon or on Mars. So it's very nice that we can cultivate cyanobacteria, use them as biomass to feed the bacteria that can in turn produce compounds that are needed in a biological life support system to support human outpost. And we are doing this research also in the frame of the Space It Up, that is another Italian project. And the final idea, of course, is to use desert cyanobacteria not only as biomass, as feedstock, but to use this as a chassis for synthetic biology application. But for doing this, we need to know a lot of things that right now we don't know. We have to investigate the proper promoters, the proper ribosomal binding site. So there is some work to be done before using these cyanobacteria as a chassis for this kind of application. But we are also trying to translate the radioresistance of desert cyanobacteria of the genus Chroococcidiopsis to other organisms like bacteria or cyanobacteria that can be more easily used in biological life support system, but these organisms are more susceptible to radiation. So in the frame of another project supported always from the Italian Space Agency, we are investigating the molecular basis of the radioresistance of Chroococcidiopsis strains, and we are investigating the genome, and we are investigating the transcriptomic response after exposure to ionizing radiation. And we are also giving a focus to proteins that are known to bind the DNA and to provide a shield against reactive ROS species. And we are transforming Synecocystin and E. coli with Chroococcidiopsis genes. And there is also the idea to transform lettuce to have a more a sort of superfood to be used on human outpost in the future. And of course, the possibility to enhance the radioresistance of organisms that can be used in bioreactor, like cell factories, is important, because currently there is a lot of methodologies ready to use cyanobacteria and also bacteria like E. coli to produce drugs, biomaterials, fuel, oxygen in the case of cyanobacteria. So even though at this stage we cannot use directly Chroococcidiopsis for space biotechnology or space synthetic biology, we can try to understand better the molecular basis of its radiation resistance and improve the susceptibility to radiation of other organisms that are maybe more important and more let's say, more advanced for space application. And yeah, that's all. So this is my currently my team at Vergata University, also collaborator from engineering department, astrophysics, bioinformatics, and also genomics, because we are trying to understand how these cyanobacteria can survive desiccation and radiation from, let's say, in molecular biology point of view that is going to be more, let's say, advanced compared to the very first investigation that have been shown to you this afternoon. So with this, I thank you, and I'm ready to take questions if there are.
Thank you so much, Dr. Daniela. I think that it has been an amazing presentation, especially since I think that the applications of this research can be applied to all of the things. I mean, as you have said, there is an infinite possibility. I mean, in food, in biofactories, and in such. I've got a question regarding the intrinsic chassis of cyanobacteria. So, what do you think that would be one of the one of the biggest problems if we had, like, for example, that you can cultivate cyanobacteria in Mars, but and you can forget all of the economics part because that's such a big part. What do you think that would be the most one of the main issues specific of cyanobacteria that maybe other species don't have?
For synthetic biology, do you mean? Yeah,
Yes.
yeah. But first of all, is the possibility to regulate the gene expression. So a lot of promoters that work very well with E. coli, they are not working that well with cyanobacteria. So we used a couple of promoters that are currently using E. coli, but we could not have the repression, so they were keeping working so they don't get repressed as in E. coli. And also ribosomal binding site should be investigated to have an overproduction of the protein that you want to produce. So we did some things because, as I show you, we did this luciferase based SOS DNA damage system. Um, and we tried a couple of ribosomal binding site, but we did not do it systematically. So this needs really to be done in a very systematic way, maybe to find the best one. And in this case, for the SOS DNA damage plasmid, we used an endogenous promoter of the RK protein. So this was easy, let's say. But a lot of work has to be done if we want to really to have a chassis. We need all the right biobrick. But good things is that generally it is not easy to manipulate cyanobacteria. There are not so many plasmid that work with everything. But desert cyanobacteria, most of them, not all, can be transformed. I have in the laboratory plasmid with an origin of replication that it's working, and it is being obtained by an endogenous plasmid of Filamentus cyanobacterium. So this also a problem, the possibility to make manipulation, to have plasmid that can be maintained, and also if you want to do the integration in the genome, then you have to identify neutral site where to be used as platform to integrate whatever you want in the genome. So really a lot of work needs to be done. And yeah, currently I don't have a project on this, but should be done definitely.
Thank you so much. I don't know if Sophia has got a question because I've got another one.
Yeah, yeah, yeah. Usually you can do the electroporation, but you have to wash to remove all the polysaccharides, and sometimes triparental mating with E. coli it's more successful. Yeah, yeah. We grow regularly with the medium that is just water and few salts and fixed nitrogen. But if I have to say the other side of the nice things is that they grow very slowly. So the generation times is few days, and so you need the months to have biomass. So this is really maybe a problem for biotechnology. Synecocystis is growing much more faster, and other cyanobacteria are growing much faster, but they are not so resistant. So you you cannot have all. And also having this thick envelope and a lot of isopolysaccharides has made also very difficult to extract genomic DNA, very difficult to extract RNA for transcriptomics. So we are, but we did it. I had a PhD student who actually has been very successful in doing this, and we are now submitting a paper on transcriptomics in response to far-red light. And we recently also published a paper on proteomics because also proteins. This was easier to get, but let's say that at the very beginning, the difficult was to obtain and achieve a gene transfer, and we did it a long time ago. Then the new challenge was to apply omics to this desert cyanobacteria, no? To go to the level of publication of that we are used to read and the quality of research that is currently done. And this has been really very difficult. We really spent years, literally years, to have this done. But now I hope that we can contribute better to understand desiccation and radiation resistance.
That's so cool. Thank you. I guess this also slower reproduction helps them to be more resistant to all these things. So
Yeah, of course, because if you go slowly, your metabolism is reduced, as I think, and yeah.
very interesting. But I'm gonna let other people ask.
Okay, so I've got a question regarding I mean, lots of cyanobacteria are holobionts or symbionts with other organisms. For example, Prochlorococcus is symbiont with Trichodesmium and such. So I was wondering if have you considered for your research, or if it's an idea or if maybe it's a concept that you have been doing research on maybe engineering or doing research also on the holobions, so maybe they can give the cyanobacteria
Mm.
that you are looking for nutrients or secondary metabolites that maybe they need.
Yeah, so this culture collection is not taxenic, this is what you are asking, and there are bacteria growing deeply in the polysaccharides, and usually we so eliminate the reeds that are not of cyanobacteria because they have a different GC content likely. And yeah, of course, it would be very important to understand this kind of association because in nature they have a role, no? An important role also of exchanging of materials, information, metabolism. But it's something that I didn't investigated, but it should be because communities are more important for survival than single species, no? That's for sure. Yeah.
Thank you so much. So there is a question in the chat. It's a okay. So it's from Maria. So she's asking if, considering the structural and protective requirements for cyanobacterian biofilms, my question focuses on mineral enrichment strategies. So she says that beyond basic nutrients, what specific trace metallic elements do you identify as critical for modulating the EPS matrix chemistry to
Mm.
enhance radiation resistance?
No, really no. But growing, I mean, I did not investigate it in detail as it is asked, but I know that I mean, we noticed that when we cultivated these cyanobacteria with the regoliths, and also when we were simulating the Mars condition, we you know that there are also perchlorates that are toxic because they induce osmotic stress and oxidative stress. There was an abundant production of EPS, so this for sure. And yeah, so the medium was the basic. I mean, when we use regoliths, Moon and Mars simulant, it was like the composition as it is sold by companies, and we added only nitrogen, fixed nitrogen, because these are not able to fix nitrogen, so we use the urea maybe to provide this source. And we added perchlorates to better investigate also the effect of perchlorates. But yeah, we did not investigate, not even at the molecular level, all the genes involved in the EPS production. But for sure, when they produce biofilm, we saw that going at the confocal microscope that a lot of APS are produced because they keep the cell attached to the substrate and each others, you know? So yeah.
There is another question from, okay, so they say that from an ISRU perspective, so in situ resource utilization perspective, how can we engineer these biofilm mineral interfaces, like the unions, to effectively mobilize and sequester the essential metals that they need from potentially toxic planetary regolith? So I think that amplifying it. So do you think that there is a way to engineer the biofilm so they can take the metals without without being intoxicated with maybe particles that the moon and Mars may have? I think that that's more or less the question.
But well, for what we did using the regoliths of Moon and Mars, we didn't see any toxic compound.
Okay. Hmm.
So they were growing nicely, and we even added the perchlorates, and so no toxic effect that we have seen. And we also use a lot of increasing concentration of perchlorates, simulating different location of Mars, and so we are still in the range of the concentration that are present on Mars. And also the moon regoliths, the composition was the one that is simulating the moon, and they grow nicely.
Hmm.
So maybe there is need of fixed nitrogen, and yeah, they grow nicely but slowly. So I have to say that the produced biomass is reduced compared to the regular medium. But if you
Okay.
want just to use water and soil,
Okay, so that was like my next question, that if you if you had seen differences in growth between the normal Earth conditions and outside of Earth
yeah, because
conditions.
yeah, this the mineral that are released from these regoliths are not as the BG11 medium, the standard medium that we use in the laboratory. So
Hmm.
the growth is supported less, so you get less biomass.
Okay. Okay, so thank you so much. I don't know. I haven't seen any message else in the chat. I don't know if anyone else has got any questions, Sophia, or I think that we are also over time and we don't know we don't want to to take more of your time. I mean, it has been an incredible honor having you here today. I'm a great fan of your work,
Thank
especially
you.
because of the interactions of living beings with the chemical compound of outer world.
Yes.
So thank you so much for being with us here today. It has been an incredible honor. It's a dream having you here today, and hope to see you and to hear from you very, very soon.
Oh, thank you. Thank you very much. And best luck to all of you for your research and the things that you love to do.
Thank you so much.
Bye.
We'll see you very soon next week in our last week. We'll see you in next week in our next lectures of the European Societies of Synthetic Biology on Thursday with our next events. So see you all very soon, and we're close the third week of the festival. So see you very soon and see you on Thursday.
See you.
Bye. Thank you.